introduction here for Dr. Cochran, Kimberly Cochran. She completed both her PhD and Master's of Science degree in plant pathology at the University of Arkansas, Fayetteville, Arkansas, where she's originally from. After graduation, she accepted a job with Texas A&M and moved to Uvalde in 2015. Dr. Cochran's office is based in Uvalde, located within the Texas Winter Garden area. And if you're not familiar with that, she'll tell you if you really want to know. It's southwest of San Antonio. I had never heard of it. Her district ranges from east of Austin, past Bastrop, Texas, all the way west to the U.S.-Mexico border at Del Rio, and covers about 30 counties. The Winter Garden, Hill Country, Austin, San Antonio, and surrounding areas grow a wide variety of plants, including row crops, commercial and homegrown vegetables, and specialty crops like olives, grapes, sesame, and pecans or pecans if you're from New Orleans. In addition to providing educational programs and diagnostic support, Dr. Cochran currently has projects working on spinach, cabbage, sesame, and olives. And I introduce Dr. Kim Cochran.
Hi, guys.
Everybody mute your, and you can go ahead and turn off your video.
Yeah, okay. All right, so I think the best way to do this is probably just kind of jot down your questions until the end, just because whenever I'm in presenter mode, it's hard for me to see the chats. So if you guys have questions, please, again, just kind of jot them down and we'll get to them as we finish up. Okay, so today I'm going to talk to you about plant diseases. So we'll go ahead and get started. All right, so hold on one second here. Let me double check. Okay, so can everybody hear me okay?
Yes.
If you, okay, perfect.
Yeah.
Okay, so first we're going to talk about plant health and plant disease and what those things are. Then we'll talk about disease management, plant disease symptoms and signs and some examples, plant pathogens and disease examples, disease diagnostics and exercises, and then we can go over some resources and other info if we have time at the end, but that is in like a home reading section. Most of that does not require a lot of explanation from me, but I do like for you guys to have some of those bits of information. So, all right, so there are a lot of definitions for plant health. And in a nutshell, though, it's when a plant is free of disease, stress, or injury and can reach its full potential in terms of yield or other production measures. That may be cut flowers, could be bushels per acre, could be pounds of lint per acre in the case of cotton. So it's really just what are you interested in from that particular plant when you're considering its full potential. So why is that important? Well, some of the obvious things are maintaining our food security and stability of our food production systems. This includes supply chain stability. That's been a major issue lately since COVID started. And also having safe food to eat. In the Western world today, that's generally not a major concern until we have a foodborne illness outbreak. But generally, we're not concerned about things like ergotism in our grain or lack of food or things like that. Also, it's important for producers to maintain profitability of crops, and that includes direct costs of disease management to farmers, as well as a yield loss, both in quantity and quality, because farmers do get a lower price for their products if they're poor quality, and that can happen with a lot of diseases. All right, so what exactly is a plant disease? It can be anything that disrupts a plant that interferes with its normal structure, function, or growth, and leads to the development of symptoms. Now, whenever we talk about disease conversationally, a lot of people mean all of these things in this graphic right here, including disorders and injuries, which are kind of more of non-living issues, or non-living causes, rather. And that includes things like air pollutants, chemicals, soil issues, nutrient issues, and several other aspects. Now, as a plant pathologist, I end up getting samples that cover all of this right here, but this area here in the green circle is what I really focus on. These are things that are living organisms. That's up for debate in the case of viruses, but we call them, we put them in the living category, and we basically kind of use that lingo with plant, like within our plant pathology field. So when a plant pathologist says a plant disease, this is what we're meaning here in the green circle. And we would call these a disorder. Though in casual conversation, most people call it all a disease. All right, so what parts of the plant can be affected? Unfortunately, all of them. The top parts, like stems, leaves, fruits, flowers, as well as roots. We have two main categories. Those are soil-borne versus foliar pathogens. And those are loosely named based on the area of the plant that is initially infected, and also how that pathogen survives when it's not infecting another plant. Types of pathogens that we'll focus on today include fungi, bacteria, viruses, and nematodes, with a main emphasis on fungal pathogens because that's most often what I come across. Okay, now that we've talked a little bit about what disease is, what is required for a plant disease event to happen or an epidemic to happen? Well, we have this thing called the plant disease triangle, and it should be in your books, but it's just a kind of a concept that we use to visualize the three main things that are required, and that's a favorable environment for the pathogen, a susceptible host or plant host, and a pathogen that can cause disease on that plant host. And some more advanced or newer texts rather make this a pyramid, and then they include time. But for the purposes of our class, we could just look at it as a triangle. So whenever we manipulate one or more points of this triangle, then we no longer have a disease epidemic occurring. So we can alter the environment, we can change up the host, replant with a resistant variety, for example, or manipulate the pathogen in some way. So this can be a really helpful tool to visualize opportunities to manage diseases by disrupting the disease triangle. All right, so a very basic generalized disease cycle here. This is just to kind of show you the general life cycle or disease cycle of any given pathogen. So most pathogens will overwinter or kind of have a state of being at rest. And this can occur in plants and plant debris, like living plants or dead plants. It can occur in the soil. And once conditions are favorable and a plant host is available, the pathogen will then attempt to infect the plant. And if it overcomes plant defenses and gains entry, then infection actually occurs. After that, the pathogen will continue to colonize the host plant, and through a series of metabolic interactions and molecular interactions, the pathogen will start to cause symptoms in the plant. And this is usually when we'll start seeing problems pop up. So you'll see things like leaf spots or wilts. And after this, the pathogen will start reproducing and spread. Now, some pathogens have a secondary cycle within a single season. So some pathogens can go straight from reproducing and spreading back to finding another plant host. And you can have multiple cycles of this within a single season, while others only have one main cycle during the year. And I like to include this because when you understand a specific pathogen's disease cycle, you can really kind of, with some practice, better understand when you can intervene to stop this cycle of disease. So, you know, on our last slide, we looked at the things that we can manipulate to stop a disease epidemic, a plant host, the pathogen, or the environment. And this shows you the disease cycle and kind of gives you insight into opportunities of when in the cycle you would want to stop it. So let's say I have the start of a plant disease problem in my garden, and I would want to stop it right here. That would be ideal, rather than waiting to here or later, especially in the case of diseases that go through multiple cycles in a season. So that's important to keep in mind in terms of timeliness of plant disease management. All right, so you guys are actually my first class with this new template. I used to have disease management later on, but I've moved it and switched things up, so you are my guinea pigs. All right, so we're going to talk about disease management strategies. The first being, oh, here we have chat. Okay. Can you guys see the chat box? Is that in your way at all? Does anybody have the ability to unmute themselves right now?
I can see all the slides, and the chat box I can move if I need to.
Okay, but you're not seeing my chat box. It's not in your way.
No,
right? Okay.
no, you're good.
Just checking. Okay, just wanted to make sure I wasn't blocking like a quarter of your screen. Okay.
No, you're good.
All right, thanks. Okay, carrying on. So resistant or immune plants are always the first thing that I recommend. It is by far the front line of defense, if you will. We also have avoidance through cultivation techniques and exclusion of the pathogen, protection, eradication, and therapy. There we go. So these first four are focused on prevention. These last two are focused on treatment after the fact. So what exactly is resistance and immunity? Well, resistance is when you're employing plant genetics and traits of the plant to naturally resist pathogens. So here's a case of pepper, and this plant on the right has Phytophthora blighted pepper, and the one on the left has been exposed to it, but because it has genes for resistance to the pathogen, so it's able to have metabolic activity that prevents successful infection by the pathogen, then it doesn't have disease progression at all. So even though these two plants were treated the same, they're exposed to the same inoculum, you can see the one on the right is faring much worse than the one on the left. So that's a really good example of how easy it is to manage disease using resistant plants. So what I just described is a good example of direct plant resistance, where the plant actively prevents disease using genetics. Indirect plant resistance is when plant traits like a thick waxy cuticle can prevent pathogens from gaining entry, for example. But with indirect resistance, those traits will have other purposes as well. So its sole purpose is not just to prevent disease. So in the case of a thick waxy cuticle, that's really beneficial to prevent sun scald on leaves, especially in some tropical plants or desert plants. It also helps reduce water loss through respiration. So we have, again, two types of resistance, direct and indirect. Though 90% of the time or more when you hear someone talking about, oh, well, this variety of plant is resistant to, in this case, Phytophthora blight, they're referring to direct resistance. Again, it's your best option. It is by far the least effort, and it's usually really cost effective. The downside is that we don't have resistant varieties for every pathogen out there, and we don't have resistant varieties in every crop. For example, in olive, it is very susceptible to cotton root rot, but we don't have any resistant varieties to cotton root rot in olive. That's actually a project that I'm working on. So that's an example of, we would love to have that option, but we just don't right now. And that's one key area of plant breeding programs where scientists are always trying to advance that and get more resistance options for disease management. Okay, so avoidance is avoiding disease development by making the environment unfavorable for the pathogen using cultivation techniques. And that includes adjusting your irrigation method and timing, the use of mulch to help reduce splashing and tillage, And tillage helps basically turn over that plant material that could be harboring disease. It helps it decompose faster, but it also just removes it from the surface of the soil. Also, different or altered planting sites to less favorable areas for disease, like drainage. If you look here in the middle, we have some pretty sad looking tomato plants. That's not a very good site. If you have a spot that is constantly backing up with water, obviously you can swap things up to move them to a better drained site the following season. Also, you can do things like alter your planting times to avoid favorable seasons for various pathogens that you know you've had a problem with in the past. And also use varieties that are adapted to your area to avoid stressed and weak plants. Stressed out plants are always going to be more vulnerable to disease. You know, a lot of people move to South Texas and they have a plant they love from where they moved, and they want to plant some cuttings or something, and it's just not suited for the area and it never does well. So always try to use something that's well suited for your area and time of year that you're planting it. Okay, exclusion is employing methods to keep the pathogen out. This includes only using healthy plants, buying certified clean plant materials, maintaining clean equipment, and having infrastructure to maintain a clean growing area. For homeowners, this really includes things like having just all your sanitation supplies set up and ready to go, because if you have that ready to go, you're more likely to follow through and use it. Like disinfecting your tools with a 1 to 10 bleach solution, for example, and then following it up with some, you know, tool spray or WD-40 or whatever your spray, your application of choice is to prevent rust. So the key here is cleanliness and being very careful with what you bring into your garden, and just be mindful of that. The limitation is you can't control everything, especially with diseases that are airborne and can be blown in on the wind. So, you know, we just do the best we can. And it usually does at least make a little bit of a difference there. All right, so protection is utilizing a barrier to provide the plant, or to protect the plant from the pathogen. So this is for use before you have an infection problem or before you have a massive disease epidemic on your hands. And with this, we can use chemical barriers that kind of go on as like a little coat to protect the plants, biological barriers, or physical barriers. So biological barriers, those are a lot of your biofungicides or biological control fungicides, and they can act a couple of different ways. Some of them colonize the surface of the plant and don't allow the pathogen to essentially get a microbial foothold in terms of habitat, or microbial habitat foothold, I should say. Others actually act more directly on the plant and kick off plant defense mechanisms and sort of prime the plant defenses. So, you know, those again are considered a barrier method though, because of the way they're applied. And physical barriers are as simple as like a raised bed or box or container gardening. So in a nutshell, you're protecting the plant from infection by the pathogen in some form or fashion. And
Can I ask you a question?
Yeah.
So is this something that you routinely recommend just to people like myself, that I just have a yard and I don't really think about disease coming into my yard commonly? Is this something that you would do, or do you worry about resistance in these pathogens, or?
That is a good question. On the protection one, and this is one of those nuanced things, with the protection methods, I recommend people really only use, especially the fungicides, when they either had a problem with it last year, so you have a known history of that problem going forward, or it's something that is just so prevalent in your area, you know you're going to have a problem with it like every year. Like for example, here in the winter garden on spinach, we know we're going to have a problem with downy mildew every single year without fail. So producers will preventatively spray various products, including fungicides, to protect the plants, because protectant fungicides are far better for food crops than systemic ones. So even in commercial production, you're not going to have a systemic fungicide easily get labeled for a crop that we consume, right? Because the protectant ones eventually wash off, or can be washed off with soap and water, for example, versus you know, if it's systemic and going all throughout the internal tissues of the plant and you're eating the leaves of that plant, then that's not as ideal. So anyway, basically, if you know you have a history of a problem, that's when I recommend the protective fungicides.
So if you don't think you have a problem, you don't necessarily need to do this as a preventive measure.
It really just depends. I mean, usually no, but there's no harm in doing like a raised bed, for example, you know, especially if you have a couple really high value heirloom tomatoes that are very susceptible to every disease out there. I would say maybe consider putting them in a really nice raised container or bed or something. You know, but it's kind of also a judgment call on value and investment and things like that. You know, like if you have some prized cuttings that are very rare, I would suggest going ahead and doing the protection ahead of time, versus if it's just run-of-the-mill cucumbers that you bought from Lowe's, then I wouldn't worry about it as much. So,
Okay,
and
that makes sense.
yeah, and this can also be useful for once you have noted a minor problem. And again, this is one of those nuanced things, and it protects from additional infection, but that only works if you ruthlessly call out any infected plants or visibly infected plants and then spray on schedule perfectly the remainder of the season, which a lot of people really struggle to do due to cost or just time investment. All right. So again, this is focusing on preventing disease by protecting your plants from initial infection, and they're not helpful after you already have a significant epidemic on your hands. All right, eradication. This is for once you already have a known problem with a pathogen. This is where you kill or get rid of the pathogen, and this includes sanitation efforts like removing or destroying diseased plant debris. And also things like when you have a case of a soil-borne pathogen, you can solarize the area. Used to, people would fumigate on commercial scales, but very few people do that anymore. People do still try to biofumigate with things like brassicas, and I actually have a handout on that if you're interested. But basically, in a nutshell, the solarization works on the principle of heating up the top 6 to 8 inches of soil to a temperature that's hot enough to kill most pathogens. It is also good for eliminating or greatly reducing the weed seed bed when you're preparing new sites for a garden. All right, so therapy is helping the plant to heal, and this is always most effective when paired up with things like pruning out the old diseased tissue, like in this picture here. So this is often going to be something that people with a pesticide applicator's license would employ, and it focuses a lot on systemic chemicals paired with that removing of infected tissues. And it's usually going to be something that people do for trees or other more permanent plantings, because honestly, it's usually more cost effective to just replant annuals, depending on what your disease problem is and the cost of that treatment. There are a few like Daconil, it's not horribly expensive, and it is useful for some garden plants, but you know, that's just a personal finances call. So. All right, so disease symptoms and signs. This is where we're going to get into the meat and potatoes of the presentation on the biology side of things. So what are symptoms versus signs? Well, symptoms are changes in the plant in response to a pathogen like wilting, yellowing, tissue death, stunting, and galls. So signs are direct evidence of the pathogen, like actual pathogen tissue. Typically, you'll see fungal tissue, or sporulation, which is reproduction of the fungal organism, or bacterial ooze. You will never see sign of a virus because it is too small. Typically, you won't see sign of a nematode unless you have some microscopy tools and skills at your disposal. All right, so here we have a cabbage leaf, and the symptoms would be yellowing or chlorosis, tissue death or necrosis, and wilting. The signs, if you look closely here, this photo is just this little part in the middle, zoomed in. That is actual fungal tissue, those little black specks and the white cottony material on the surface of the leaf. So the black specks are reproductive structures of the fungus, and the white cottony material is the fungal somatic tissue, if you will. And we'll talk more about those in detail in a few minutes. So symptoms are what happens to the plant in response to the disease or to the pathogen infecting it, and signs are actual pathogen tissues. All right, so what are some common symptoms? I see a whole lot of yellowing and dead spots, especially leaf spots and blight. Leaf spots and blights usually occur due to fungi, bacteria, and viruses. General yellowing, though, can be nematodes or nutritional disorders, for example. Here's another example of some leaf spots. And notice here that we have a tomato and a peanut plant. And both of these are actually infected by the same type of fungus. So this is a really good example of some fungal pathogens have a really broad host range because, you know, tomatoes and peanuts are not really closely related. They're in different families. A tomato is a solanaceous crop and peanuts are legumes. All right, so here we have a very different looking manifestation of some of those symptoms. So here we have yellowing or chlorosis progressing to dead leaf tissue here. And this is pretty typical of oak wilt. And starting from the veins going outward, it's one of those kind of red flag symptoms that we look for in oaks in the live oak family. And I really like to include this kind of broad spectrum of how these symptoms look, even though in our verbal language and written language, they sound the same. So, you know, yellowing, chlorosis, dead tissue, well, that applies to all of these pictures, but they look very different. So, you know, sometimes with plant disease identification, pictures are your best friend. And also I've included some little vocab tip boxes here and there. So those are just little bonuses for you. All right, so some more common symptoms include cankers and dieback. These are often fungal, sometimes bacterial causes. I see this a lot on roses, like so much. So here's a case, a pretty severe case of dieback on rose. See here, it's almost made it all the way down to the crown. Here's a close-up of what that looks like. Here you have the canker line. And people always want to know, well, you know, can I get rid of this diseased tissue and save my rose? Because sometimes it's a pretty big investment, or it's, you know, just a favorite one you've had for forever. So what I would recommend is pruning further down, like at least 4 inches below if you can. I know sometimes it's impossible if it's close to the crown, like over on the picture on the left. Basically just prune at least 4 inches or as far down as you can, because the fungal tissues in this case, I believe if I recall correctly, this is Phomopsis dieback, but the fungal tissues on this are not just here. It goes down here. It's kind of like an iceberg, so you're only seeing part of what's actually there. So always cut lower than you think you will need to, and a nice angled cut will give you the best results with that. All right, here's another example of dieback on a tree. I see this a lot on trees. And on this one, this is actually caused by a bacterium called fire blight on a pear. But around here, for the most part in Texas, well, south-ish, southwest Texas, I usually see fungal issues that cause dieback. But anyway, this is a really good example, though, of where it's progressed down from the twig into the main branch. So again, we would definitely need to prune way further back down on that branch to try to prune that out as best we could. I also do see a fair amount of galls, which are these kind of tumor-looking overgrowths. And these can occur above ground and below ground. They can be caused by fungi, bacteria, or nematodes. And, you know, for the most part, people always talk about agrobacterium that causes this. It causes crown gall. Nematodes, like root knot nematode, can cause some pretty dramatic looking galls. But, you know, when you see above ground galls, like on trees, like burls, I often find those are caused by fungal pathogens in this area. That's not to say there are no other reasons for that on trunk tissues of trees and other woody plants, but that's generally what it is.
So would you treat that with a fungicide, or would you just get rid of the tree?
Get rid of it. Well, well, let me rephrase that. It depends. That's the thing with trying to manage living things. It always depends. It depends on where it's at. If it is on the trunk, you need to just get rid of it because you can't cure that out of the trunk, and it will slowly decline. But the thing is just like how fast will it decline? You know, and it is really dependent, you know, like how long you want to leave that there is dependent on how close is it to structures, is it a danger to people, and things like that. So there are a lot of factors you need to take into, you know, into account before you make a decision about that. But actually, at the very end of your presentation in the kind of like bonus slides, there's a little slide on burlwood, and you guys can take a gander at that if we have time. Well, you can on your own time, but we'll go through it for class time if we have time. All right, and just to try to speed things along because we have a lot of material, let's try to jot down our questions and save those for the end. All right, so some more symptoms that I see a lot are root and crown rots. And I see these a whole lot on seed starting and seedling operations. And here's on the left, an example of white healthy roots. In the middle there, we have some really severe damping off. That's pretty typical of advanced or severe damping off. Notice the roots are all discolored and the whole seedling looks fairly water soaked and limp. That thing is definitely a goner. Now on the right panel on that little trio right there, notice that we have a seedling that has a little more root mass than the severe one, and it's not as discolored, and the above ground tissues seem like they're getting along okay. And this is a really good example of the fact that plant diseases are not always an all or nothing thing. So, you know, you might have a moderate case of something and never even know it. And the plant will go along okay, but it might not ever reach its full potential, but it, you know, it's not dying either. So that's just something to keep in mind, is that it's not fully black and white. All right, so also I see a lot of crown rots, particularly in ornamentals and houseplants. And this is often associated with overwatering. Some people think it's just because you overwater, but it's usually not. It's actually usually because you have a pathogen there that becomes active when you overwater. And that's linked to that disease triangle. Remember, when we have a favorable environment, that's when you have a disease problem. So what usually happens is you didn't fulfill the disease triangle requirements until you overwater. So that's why you often have a problem, or don't have a problem until then. Also, seedling disease and root and crown rots are usually caused by fungal pathogens, sometimes a complex. I run into that quite a bit. Sometimes that complex can also include nematodes, and sometimes I do see bacteria with this, but not super often. I have only come across that a handful of times in this area. All right, so some plant pathogen and disease examples. This is the really juicy stuff with lots of pictures. So we're gonna save the fungal group for last because it's the biggest one and my favorite. So we're gonna start out with bacteria. And these are single-celled organisms. As you guys know, they're everywhere in the environment, but only some of them are pathogenic to plants, just like only some bacteria are pathogenic to people. There are a lot out there that don't bother us, and there are a lot that don't bother plants. These include things like leaf spots, wilts, scorches, soft rots. Those are all really common. And most that cause disease in plants will not make people sick. However, it can if you are immune compromised, and you can find more at that link right there. I feel like I've now done my social duty for the day. I have shared that little health related bit for y'all. It is important to note though, that some bacteria that are pathogenic to humans can survive on plants, like E. coli, Listeria, and Salmonella, and many others. So just because, you know, most plant pathogens won't make most people sick, it's still really important to have really good cleanliness practices with your harvest from your garden, especially if you're selling that at market or sharing it with others. So up here I mentioned the wilts. That's one of the more common things that I see in terms of bacterial diseases. So here's a tomato stem that's been cut, and there's bacteria streaming out, this white ribbon. That's bacteria by the millions that's just pouring out of its vascular system. And where's my video? Hold on one second. I will be right back. I need to find my video. It has escaped my presentation. So give me 1 second. I promise it was there last night. I'm just having to go into my mains folder and find it. I'm sorry, my computer is so slow, like really slow. I don't know what it is about today being super duper slow. So while I'm looking, does anybody have any questions? So we don't end up wasting our time as I'm desperately trying to find my stuff.
I had one earlier. I put it in the chat. You were talking about diseases splashing up from the soil, and that one of the solutions is to mulch. But I have a slightly different problem where I live in an apartment complex and I'm a container gardener, and the lawn crew weed whacks really close to my plants, and they kick up dust and dirt into my plants and transfer soil from the lawn into my plants. So I can't exactly mulch the lawn. It's supposed to be lawn. Do you have any solution that I can somehow put a barrier between my plants that are already in pots that are 18 inches high?
Honestly, with that, that's just kind of, yeah, that's a difficult situation. If you could just move them inside, I know that might be difficult given the size of the pots you described. Another option would be just to like essentially erect a little barrier there just for the day, you know, like a little tarp or something. That might be an option. But yeah, that's unfortunately one of those things that if you were doing your own, if you had control of the lawn work being done, it wouldn't be a problem. But yeah, just short of erecting like a barrier, there's not much I can think of really. Though, I mean, if it's lawn and it's just blowing dust up, I don't think it would be as much of a risk in terms of they're not blowing up just straight, you know, like plant debris from vegetables or something that's closely related to what you're growing.
It's grass clippings, it's weed clippings, and it's soil, because they literally like weed whack down to the dirt level. And yeah. And it's Victoria clay, which is full of cotton root rot.
Yeah. If they're weed whacking down to the dirt level, there's not much you could do. I would just put a little tarp barrier up on the days they're going to do it if you can, because there's just not much else you can do. Okay, well, I got my video.
Awesome.
Okay, all right. So I finally found it. I guess the file size was too big for my presentation. So this is a tomato stem that is in a little beaker of water, and this white stuff streaming out right here, that's bacteria that's streaming out from the stem. Now this is only really something that we can use as a little quick diagnostic screening tool for bacterial wilts. This doesn't work for leaf spots. It doesn't work for things like citrus greening. It only works for bacterial wilts. So just a quick little replay for y'all. So this is not sped up, that's real time. This is a really severe infection. So in other cases, it might be just a few little tendrils But when it's against a dark background, notice here they put like black paper behind it, then it's pretty noticeable. So, all right. So now I'm going to share my presentation screen again, figure out how to do this. So normally, we have a question about normally there would be nothing streaming after cutting. There might be a little bit of sap, but it's not going to look like that unless you have just some like really milky sap species like a milkweed or something. But even at that, it won't look quite like that. It would just be more like a general seepage, not active streaming. Does that make sense? Perfect. Okay. Alrighty. Okay. So a little more about bacterial wilts. Here we have some pictures of bacterial wilts, particularly on tomato and a cucurbit. So with these, we always see a water-soaked limp appearance, and they almost always result in eventual plant death. Here you can see that really water-soaked limp appearance on the cucurbit. I believe that's zucchini. Early stages, before it looks this severe, will be most apparent at midday when it's really hot and the plant just cannot take up water, even though you've watered it. And it will look wilty despite like as much water, or despite watering it as much as it can stand, it will still look like it's under drought stress. Well, bacteria in general can often spread easily by water splash and through wounds. So we focus on preventing insect injury. That creates a lot of little entry points where bacteria can get in. Bacteria also get in through natural openings. So even in the absence of injury, they can get in through like stomata and hydathodes and things like that. Reduce your water splash, especially from plant to plant, and try to prune things out so it's thinner, particularly when you have a lot of closely planted tomatoes, and that will help reduce your canopy humidity and reduce the favorability for the pathogen in that environment. Cull infected plants. You don't want a typhoid Mary sitting there infecting all the other plants nearby it, and practice good sanitation when you do. So don't just pull it up and leave it there on the ground. Pull it up and dispose of it properly. And then wash your hands after you do that before you go handle other plants. Rotate crops for a minimum of 1 year, and be on the lookout for no volunteers or weeds, because those can be bridge crops and infect, basically carry over infection to the subsequent year. So here you can see the tomato that's been infected, and when you cut the stem lengthwise like this, you can see that really chocolatey discoloration, and it actually is missing some tissue in the vascular tissue right there. That's a key red flag for bacterial wilt. And sometimes verticillium wilt can look like this, but usually verticillium wilt will look a little more black than brown. Bacterial wilts, to me, usually look more brown in the case of vegetables. And also if you cut it the other direction, so longitudinally, then you'll still see this kind of brown discoloration in some of the vascular bundles. So that's another little quick tip on checking your plants or checking your sickly plants for bacterial problems. Okay, one other quick screen that I forgot to mention is a little more advanced. It's a little more difficult, but you can actually take an affected or suspected to be affected plant and cut it lengthwise across the stem, and then very carefully put it back together, and then slowly pull it apart. And if you have this really sticky gummy substance kind of like clinging to both ends and forming this string, that is really a good sign that you have a bacterial wilt problem. This is going to be far more slippery and sticky and kind of mucousy than sap. Sap will be much thinner than this. And you can do this with tomatoes, cucurbits, most of your garden vegetables that you suspect might have a bacterial wilt problem, which for us is usually going to be tomatoes and cucurbits. All right, so another major bacterial problem that I see a whole lot is citrus greening. And this is kind of, unfortunately, very relevant to you guys. Just last year, you were included in the quarantine area for the coastal bend. And citrus greening is a bacteria, or it's caused by bacteria that infects the phloem of citrus trees, and it does eventually kill the tree. It's caused by Liberibacter bacteria. It used to be thought that it was a couple species. Recently, the clinic put out a fact sheet saying that it was, I believe, Liberibacter, I want to say, I'm not even going to say it, I'm going to butcher it. It's ASCIUS, I think. So, but I can send you the fact sheet later if you want it. It's also known as Huanglongbing or HLB disease. That was actually one of the first names that it was known by, but here in the US, people often just refer to it as citrus greening. It is vectored by the Asian citrus psyllid, and these are super tiny. You can see up here in this top right picture, that is a person's finger, and it's zoomed in so much you can see their fingerprint. So these are like the size of a pinhead or the tip of a toothpick. And all the photos here are, unless otherwise noted, from Citrus Alert. And this is actually a really good website with lots of pictures about citrus greening and a couple other issues. And I know some people from A&M help kind of do quality control on that. So it's a pretty good resource. Now, that little psyllid looks like this up close. And one key red flag for looking for these in your citrus is that they always sit at this distinctive 45-degree angle with kind of like a very flat head end, if you will, that they keep oriented parallel or against the leaf surface. So that's kind of a key thing to look for with those. And there's also some photos on the Citrus Alert website on the frass and things like that to look for from these Asian citrus psyllid. This can also be spread by humans moving plant material, even budwood and leaves. So that's one of the reasons quarantine is very important with this, is we don't want to move that material around. So here I have this beautiful little cartoon. Go with me here. It's my art. I did not major in art. Let's just put it that way. So here's our little psyllid, and it has bacteria in it that it's ingested from an infected plant. So that bacteria can multiply, and then as that little psyllid goes to a new uninfected plant, it uses its little mouth parts to feed on the phloem of the vascular tissue, and the bacteria is transmitted down its mouth part into the phloem. So this is a phloem limited infection, and the bacteria actually will multiply and colonize the phloem of the plant. And here, this is a cross section of the vascular tissue, specifically the phloem, and here you can see the holes right there, just like here. So it kind of corresponds with that photo. And here in the middle, you can see this gloppy material. That's actual bacteria clogging up the phloem tissues of this plant specimen. Now this is magnified several tens of thousands of times. It was taken using a special microscope called a scanning electron microscope. So this isn't everyday microscopy, but it is a really great image of what is physically going on in the plant. So the bacteria not only clog up the phloem by physically clogging it up just with their presence, but they also secrete a lot of metabolites and like kind of some chemicals that are really detrimental to plant tissues. All right, so what are some common symptoms? Well, I think some of the most famous symptoms and easily recognized symptoms are small, bitter, discolored, and lopsided fruit. Here are some examples of those. Things like oranges staying green, not ever fully ripening, and they stay really small, and they just taste nasty. You can also have really thick pericarps and very itty-bitty little fruit sections. Oh, the name escapes me. In cotton, it's called a locule. I'm sure it'll come to me after class. Anyway, but basically you have just a very malformed fruit in a lot of different aspects of fruit quality. You can also have raised, thickened, or corky veins. Here you can see on this bottom left picture, you have very raised veins from what they would normally be on a leaf. And notice that it's kind of a little convoluted looking. And also, this is one of the most common things we see in Texas, is the asymmetrical and blotchy modeling of the leaves. So this is by far the most common symptom we see here in Texas. And notice that this looks a little different from like a nutritional deficiency because it's asymmetrical. When you kind of visually divide it along the midrib there, the left does not look like the right. With nutritional deficiencies, they'll usually look pretty close to a mirror image. All right, so with citrus greening, detection is super important, and it's a key feature of control and management, because right now there's no cure for an infected tree. I've heard that there's some work going on in Florida and some other places on trying to find an effective treatment, but that's been really challenging. Some commercial producers do spray for the vector to try to control that. But unfortunately, those little psyllids can come in on a lot of things, including shipments, and that's how it's thought to have spread to some select locations throughout the world. But early detection and action is really important to help limit the spread of that disease, so you don't have basically a, essentially typhoid Mary sitting there in an orange grove. I'm not used to doing this without a clicker. Hold on. Sorry. Okay. Okay, so after early detection, it's really important to remove those infected trees as soon as possible. Normally, they might decline over an extended period of time, but while they're sitting there, they just serve as a source of inoculum for citrus trees in the immediate area, and it can be further away than you think. We're not talking about just the ones in the yard next to yours. We're talking a ways. I want to say it's over a mile, because those little psyllids can travel a good ways. So especially in areas where you have commercial citrus grown, quarantine is very, very important. And, unfortunately, like I said, you guys are now in the quarantine area for that. So again, detection is the front line of defense, because otherwise, without it, we don't know what to quarantine. DNA screening is really sensitive. It's a great tool, and it's fairly high throughput. You can do a lot of samples pretty fast, but you can end up having false negatives if you don't sample the right tissue. So if the infection is present in, say, the upper left side of this branch on this tree in the picture, but the technician samples from the opposite side, lower area of the canopy, then they could have missed an early positive and gotten a false negative. So it's all about the sampling, and that is a vulnerability of DNA detection with that. So some labs and the USDA have put a lot of money and a lot of effort into training dogs to sniff out citrus greening, which is a really cool project. And you can read more about that. There's actually some kind of popular media articles. This photo's from the LA Times. And you can just read about that in the news. There's also some really interesting things being put out by the USDA about their program with that. It is kind of expensive to have a dog come out and screen your grove, because it's really expensive to train the dogs, but they are very, very effective. They have a very, very low threshold of failure. So it's pretty good work. Hopefully with more popularization of that method, it will get more affordable. Anyway, so if you think you may have an issue with citrus screening, Dr. Femi Alabi, or Olufemi Alabi is his full name, is down in Weslaco. He's our main guy for citrus screening, because that's where most of our citrus is down there in the valley, in that area. So if you have an issue, his contact info is at the end, or if you need to get in touch with him through some other means, if you can't get at him by email, then give me a call and I'll get you in touch with them. All right, so this is a map that I found on the Citrus Alert website. There's a link there to the most recent map. So this is a most recent map of South Texas. You can see the Houston greater area is now under quarantine. The coastal bend is as well as down in the valley around McAllen and up into Laredo. So, continued intensive scouting is going on, but I would not be surprised if eventually we aren't essentially under a state-wide quarantine eventually. Hopefully not, but I wouldn't be surprised. So when I say quarantine, I do mean plant material and the insect vector. And with the insect vector, it's important to think about quarantine with shipments. Like, for example, if someone's selling citrus for a grocery store. They have to quarantine and fumigate the pallets to ensure that psyllid vector is not in the pallet before it goes to the facilities, or before it travels, I should say. So quarantine means that things that are within the area of quarantine that are citrus related cannot leave there without going through the proper channels and proper quarantine. And that includes plant material like cuttings or even rooted trees. You know, like if you buy a citrus tree in Houston, it has to stay within that quarantine area. You can't buy, or you should not be buying a citrus tree in Houston and taking it to, you know, Del Rio and planting it. So now to zoom in so y'all can see this better. When you zoom in closer on this tool, it does include a list of the counties, so you can see that most of your coastal bend area is unfortunately in the quarantine now as of last fall. All right, on to viruses. These are submicroscopic, and I like to go from citrus screening to viruses because they do share a few commonalities, like being spread by insects. A lot of viruses can be spread by insect vectors, and those are very extensive in their variety. You know, aphids, mites, thrips, whiteflies, you know, any number of insects can be vectors. Now, some viruses can be spread by people, like on your hands. For example, if you smoke and you've handled cigarettes and then you go into a nursery greenhouse, that can be problematic because you can spread tobacco mosaic virus to a lot of ornamental plants that way. That's why a lot of commercial nursery greenhouses have strict rules about smoking. Also, some can be spread by seed. That's still kind of under investigation on what can be spread by seed and what's not. But generally, plant viruses are associated with a yellowing, loss of vigor, and death of plant in some cases. So with viruses, there's a very wide variety of the way the symptoms can appear. So here's one example of tomato spotted wilt virus. And this is on a peanut leaf, and you have this kind of bullseye pattern. It's very distinctive. And here you have tomato with tomato spotted wilt virus. So again, these are not really closely related plants, but they both have tomato spotted wilt virus. And the symptoms actually look kind of vaguely similar. So this is a good example of you know, how widely some viruses can infect plants, while others are very host specific. But I also want to point out here one thing that people are always confused by every year, and that's how viruses are named. So, you know, it might seem odd that we have tomato spotted wilt virus on a peanut plant, but it's actually not a typo, and it's not a mistake, but rather it's just a throwback of how the viruses have, how virus naming has been developed. So generally viruses are named by the host they're discovered on first, the symptom, and then the word virus. So this was first found on tomato. It causes spotted wilt, and it was, you know, the word virus. So tomato spotted wilt virus. And then later people discovered that it also infects other things like peanut. So some other virus symptoms include kind of malformed growth or epinasty, and you can see that here on the soybean leaf where it's really puckered and very misshapen. You also have some very distinctive mottling here, and that's M-O-T-T-L-I-N-G, not modeling like prancing down the runway, but mottling, and that's a very classic virus symptom. So sometimes viruses can look a little bit like other things like nutritional deficiencies, and here's one example on a bramble. And in passing, you might think, oh, well, that's just, you know, maybe some iron deficiency or something. But when you look closely, look at the patterns, and that can be a clue as to what you're dealing with. For example, this leaf in the middle, in the lower half of the photo, look at it down the midrib and then look at the two halves. It's not the same. So one side has far more extensive yellowing than the other on that same leaf. So it's not a mirror image of each other in one single leaf. And that's a good clue that it's not going to be a nutritional issue, because nutritional issues are going to be more symmetrical than that. So we had a question about, where did I see that? So if we have a tomato that looks like on the screen, should we throw it away? I mean, you can. It might have not as great of a flavor as one that is not infected, but I mean, those are used to make sauce all the time and things like that, or cooked down in tomato paste. So it's harmless to eat. And most people actually already have let me rephrase this. Okay, most people already have the genomic material of over 20 plant viruses in their gut microflora at any given time. Now, granted, it is inert and not viable at that point because it's been through your digestive system. And generally, viruses that are plant infective are not going to survive a mammal digestive system. So, you know, you're good to go. And you've already eaten it, is the summation of that. You've already got it in there. So you'll be all right. Okay, so prevention tips for viruses. Generally, focus on vector control, prevention, clean stock. So if you're getting cuttings in particular, try to make sure you're getting clean stock, and tool sanitation and cull affected plants. All right, so a prime example of virus problems in Texas is rose rosette virus. It's spread by mites and not sanitizing your tools between plants when you're pruning, as well as physical disturbance of plant tissues, like handling and touching infected plants and then non-infected plants. Though the efficiency of spread for that method is not nearly as high as, say, mites. Mites are probably one of the most efficient spreaders of this particular disease. This always results in plant decline and eventual death. And notice on the symptoms here on the photo, there's a lot of malformations on the physiology of the plant and discoloration as well. So the thorns are the wrong shape. There's far too many. The leaves are, well, the stems are shaped poorly. The leaves and stems are discolored. They're kind of yellowed, as well as they're more red than they should be. Sometimes on some varieties of roses, you have some reddish coloration on new growth, but a lot of this growth is way too old to be having that reddish discoloration. Also, you can have an overproliferation of buds. So instead of having only a handful of buds, or even one or two, you can have 20, and they'll be far too small, and the flowers will be very misshapen as well. So prevention tips: start with clean stock, space your plants so they don't touch, sanitize your tools between plants, certainly if you have really high value roses, even between cuts, and remove any wild multiflora roses in your area, and cull infected plants as soon as possible. Again, you don't want a little typhoid Mary sitting there ruining your whole rose plot. Another thing that I forgot to touch on as I went through that list is spacing your plants so they don't touch. That is so that the mites cannot have an easy highway from plant to plant. If you space the plants, then they have to go down on the ground and then over to the next plant. So it dramatically reduces the likelihood of easy plant-to-plant transmission of the mites. All right, so tomato spotted wilt virus is another really common issue I come across, and this is both homeowner and commercial production tomatoes. It is vectored by thrips, and those can be monitored using sticky traps and addressed with insecticides if you have a problem. And again, with thrips, just like diseases, it's very important to catch those really early, because if the population gets too high, it's really difficult to get those under control. With the tomato spotted wilt virus, though, we did see that tomato earlier. Here's some more dramatic symptoms on tomato fruit here at the bottom. But you do see some symptoms on the leaves and the stems as well. Notice the kind of medium brown to light gray discoloration, as well as some stunting you can see in that bottom left photo that's highlighted with the arrow. Notice that those few plants right there are much smaller than their counterparts, and they're kind of yellowed as well. So for tomato spotted wilt virus, I always suggest remove those infected plants and weed hosts in the surrounding area that can act as a reservoir for the virus and vector habitat, and use resistant tomato varieties. That is going to be information that you can find in seed catalogs, for example, or even on tags of transplants. And also you can look it up. There are several PDF documents on the internet that have information about that, and I have some if you would like one. But with the resistant varieties, that is going to be by far your best bet. And just given where you're at, I would suspect that you guys would have a significant problem with tomato spotted wilt virus from time to time, particularly in years when thrips are really bad, like last year. All right, on to nematodes. So these are microscopic worms, and only some of them are plant pathogenic. There are a lot of nematodes out there that do not infect plants, and in terms of knowing if one is a plant pathogenic nematode or not, that is something that a pathologist like myself determines under a microscope. And we look for this special little mouth part called a stylet. Here on the bottom left photo, you can see that on the head end, which is, if you can see my mouse right here. Oh, sorry. There we go. Here's a close-up photo of that. And plant pathogenic nematodes have what's called a stylet, and it's like a little straw that they use to pierce plant tissues and suck out nutrients. So that is a very distinctive feature of plant pathogenic nematodes. Ones that are not, like the nematodes you add to your lawn to take care of insect problems, those are not plant pathogenic, so those are fine. All right, so here's a photo of a nematode that is partially embedded in root tissue, which is that kind of gray-green background in the photo. And that is a good example of how a nematode would start its feeding site. So nematodes will choose a site on roots and just start feeding. And when that happens, they start what's called a giant cell, if you will. Well, that's the actual term. So here we have some nematodes that have set up a feeding site, and they have their giant cells, which you can see here on the bottom right photo, in photo B. Those really large, they look like kind of blue bubbles. That's actually a cross-section of a root, and those large areas with the little asterisks in them, those are the feeding site cells that the nematode has developed. So basically it kind of has, well, it has these molecules in its mouth parts that hijack plant growth, and it basically tricks the plant into making these cells that have a lot of extra food for them, like sugars and things like that. So it's actually a really elaborate interaction that we could have an entire course on, but that's what makes up part of the gall itself. The other part of the gall is the egg masses. So here on photo A, there's it looks like a brown ball with little dots inside, and it says EM on it. That means egg mass. So that's an encysted female nematode that's formed a feeding site with a root, and she's actually got eggs in that mass as well. So the galls that you see are going to be part egg mass and part giant feeding cell. So they're pretty interesting little creatures that have a very crafty lifestyle with the plants in terms of how they hijack plant metabolism for their own benefit. But root knot nematode is not the only type of nematode that we have in Texas. It's the most visibly recognizable, but there are others like the sting nematode or the cyst nematode that are also pretty common. Prevalence depends on your soil type. Generally, root knot nematode, like sandy soil, though it can survive in some other soil types as well, and the same for the other nematodes. Some of these nematodes do have a very wide host range, though, like root knot nematode can infect many different garden plants, as well as commercial plants and woody plants like pecan in this photo. Generally, nematodes are very challenging to manage because there's a lack of products that homeowners can use, and nowadays even a lack of products for commercial producers to use. So we really focus heavily on prevention for nematodes, and that includes plant resistance for your replants next year. So if you know you've had a problem with nematodes, it's really important to be on the lookout for some resistant varieties for next year. Prevention of infestation, carefully inspecting transplants. It's really hard to notice anything with cyst or sting nematodes, but With root knot, if you have really careful inspection, you usually can at least do some good with those efforts. And lastly, crop rotation. So if you have a problem, you can do crop rotation, being careful of alternative hosts. So if you know you have a problem, feel free to get with myself or another pathologist, and we can help set you up with a rotation regimen that will not include alternate hosts for that disease. So you won't continue allowing that population to flourish in your soil. All right, so on to fungus. We are 5 till 7 now, so we should be okay on time. All right, so fungus is by far the most common pathogen that comes in my lab, but most fungal organisms out in the world are not plant pathogenic. There's a huge portion of them that are just decomposers that function in the nutrient cycle of your local ecosystems. There's a lot of them that are symbiotic, like mycorrhizal fungi, and they do things like increase root absorption zone areas for nutrients and water, assist with breaking down nutrition into consumable forms for plants. They also provide kind of the opportunity for a more detailed synergistic relationship with plants, where the plants provide carbon to the fungus, and the fungus not only provides that nutrition and water for the plant, but it also kind of helps sequester carbon in the soil. And that can be actually really helpful for things like sequestering carbon from the atmosphere. So in terms of global ecology, fungus actually has a really significant role, not only for helping out plants, but helping out us two-legged critters as well. So also an interesting thing is that a lot of symbiotic fungal organisms can actually facilitate signaling or crosstalk between plants. So when you have two plants that are separated by some distance and they're not connected by their own root system, if they share a fungal symbiont together, then that fungal symbiont can facilitate crosstalk with plant signaling chemicals of things like plant chemical signals for herbivory, and there's been some research even on triggering plant disease defenses that way. So it kind of gives the neighbor plants a heads up on what's going on before it gets there, so they can start their metabolic response early. All right, so on to some more pictures of fungus. All right, so the fungal group is highly diverse, and this is especially seen in the appearance of their reproductive structures, which is typically the most visible part for people. And you know, here on the bottom left, we have conch, like C-O-N-K, or bracket fungi. You see these a lot on trees. Here on the bottom right, we have jelly ear fungus. It's often seen on dead wood, especially after good rain. And up here on the upper right, we have our trusty friend bread mold. We've all seen this. And these are good examples of fungal fruiting bodies, two of which here on the bottom are easily visible to the naked eye. And then the one up here on the bread, it's a little trickier. Here you're seeing the two main aspects of fungal physiology, the hyphae and the reproductive structure. In this case, reproductive structures are microscopic. I'm going to check the chat for questions. Okay. Okay, yeah, so when we're done, I can check into the citrus bug. All right, so the two main fungal structures, again, are the mycelium and the reproductive structures. So here on this slide, we're looking at the mycelium. Collectively, it's called mycelium. Here you can see on the bottom left photo, it's really extensively colonizing this dead log. And it looks kind of cottony or thread-like. And what we're seeing is actually only a small fraction of what's there. There's a ton of it down in the log as well. And it's breaking down and absorbing nutrients from that log and helping it speed along its decomp process. So singularly, though, we would call them hyphae. And here you can see that distinction in the photo on the upper right. Mycelium is the whole thing. Hyphae is one little thread-like piece. And these are kind of like roots in that they absorb nutrients and anchor the reproductive structures. And it colonizes your substrate no matter what that may be. So this is a sign, and that is actual pathogen tissue, but it's not always visible because it may be so embedded in the substrate that you're just not seeing any on the surface. And a example of this is something we're all comfortable with, and that's mushrooms. So on this lower right-hand picture, we have the mycelium down under the ground. And whenever conditions are favorable, the mushroom itself will start forming, and that's the reproductive structure that sporulates. So you have the mycelium that's there all the time, and then the reproductive structure that's there some of the time that you actually see. All right, so on to fungal reproductive structures. These are really important in terms of me doing my job. So spores are kind of analogous to seeds in that they germinate and form a new, essentially a new microorganism. And when they're on a suitable substrate and have ideal conditions, they can germinate and colonize that substrate. And in the case of plant pathogens, that's plant tissue. So on the bottom left here, we have a photo of a rust spore. So, you know, the foliar pathogen called rust that makes those little orange powdery looking pustules on the bottom of leaves, that's a spore from that pathogen. And when it lands on a leaf and has suitable humidity for an extended period and ideal temperatures, then it can start germinating this little tube that looks kind of like a root, and that will actually gain access to the plant through the stomata. Now, some fungi can actually form these little structures that force their way down between the cells of the epidermis of the plant. But that's a different thing for a different course. We can talk about that all day. So just to keep it simple for this intro course, we'll look at how this one works. So once that little hyphae germinates and goes down into that stomata, it will start colonizing this tissue in the immediate area. And if it's successful at overcoming the plant defenses that it triggers, then it will colonize further and further and start causing symptoms. So here's in the top middle, kind of just a progression photo, if you will. We have that initial germination of the hyphae, and then it grows in branches and colonizes and eventually forms a fairly circular, typically it'll be fairly circular colony. So here, on that photo below that, that's kind of just a cross section with the hyphae dyed blue. And you can see where it is colonizing the plant tissue. The background there that's kind of like pinkish colored to grayish, that's plant tissue that it's colonized. So in the lab, I do scrapings and pick little bits of fungal tissue off of plant material and look at them under the microscope. So this right here is an example of the fungus alternaria. We looked at this earlier on the tomato and peanut. And, you know, it has these very distinctive teardrop-shaped brown spores. And here we have kind of canoe-shaped spores called, this is from a fungus called Fusarium. So these are really good examples of how diverse fungal spores can be in their shape, size, and color. And all of those features are something that I use to identify the fungus and give you a diagnosis. So you guys aren't necessarily responsible for remembering, you know, what these spores look like and how, you know, what they're called and everything. It's just a peek into the window of how things work in a plant pathology lab so you can better understand that. All right, so now that we've talked about what spores are and everything, let's talk about how they spread. So some spores are more prone to spread by wind or water splash, for example, and their physiology has a big role in that. So we have two kind of little case studies here. On the left, we have a fungal culture right here on the top left. And you can see what look like little coffee grounds with a oozy substance on top. Those little coffee ground looking structures are actually these little, they're kind of like little cups that are on the surface of the substrate that the fungus colonizes. And this is an artist's cross section of what that looks like. And these little ovals are the spores. So this ooze here, that's the spores that are being made from those cup-like structures. And it's oozing out from the cup. So down here on the bottom left, you can see embedded in plant tissue, those little cup-like structures with some little whisker-like projections off them. But from inside that cup, that's again where the spores will form and start oozing out as a little droplet. So whenever we have rain or sprinkler irrigation, you know, a droplet of water can hit this little cup shape and splash out spores and spread it wherever the water splashes. So you can see how this structure of the reproductive physiology here has a big role in how those spores successfully spread. Versus, you know, by comparison, over here looking at something we all know, a mushroom. We have a mushroom on a stalk here, and underneath or on the underside of the mushroom cap, we have the gills, and that's where the spores are made, and those drop off. So given that it's on a stalk and like held up off the ground a little bit, and you know, it's facing downward where we're not going to get a lot of rain droplets directly hitting it, I think it's pretty apparent that it's probably going to be spread by wind, right? So I have a little video here, if I can find it. It should be here. Hold on one second, I just have to cut and paste the link. Okay, so I'm sorry, my internet's so slow today. It's buffering. Okay. All right. So as soon as this plays, you should be able to see it. Okay, so here, let me find the best spot to start. All right, here we go. So this is the underside of a mushroom cap right here. This little disc that looks kind of like a space UFO right here. That's the underside of a mushroom cap. So someone has set the camera just underneath the mushroom cap on the ground. And this little bit here is just substrate sticking up. It was grown on, I think, straw. So here, all this white dust looking stuff, those are spores dropping off from the mushroom cap. Here you can see the stalk of the mushroom on the right-hand side. And again, the cap is at the top. Here's a better view. So you can see just literally millions of spores just dropping off the mushroom cap. And this isn't under a fan or anything, it's just normal air currents in a room.
I'm sorry, but I think all I see is still the slide. I don't know if it.
Okay, let me try again. Okay, can you see it now?
No, it's still on the one on how does the slide of how do spores spread.
Oh, okay, because on mine it says that it's. Let me stop share and start it again. Let me try that. Okay. Okay.
That looks like
Can you
something.
see it now? Okay.
Is it like bluish?
Yeah,
Yes.
right
Yeah.
on.
Okay, all right, thank you. Okay, so here in the middle is the stalk of the mushroom. And again, you're sitting underneath the cap. So can everybody see it now?
Yeah, and that's pretty awesome.
I know, right? And the spores are so small, it doesn't take a lot of wind to really achieve this level of movement in the video. So, you know, if you have good wind gusts, you can get some really good dispersal of fungal spores with airborne mechanisms. Yeah, and if you guys want to watch this again, the link is in your handout.
One of the Planet Earth or, I don't know, similar type documentaries did this in super high def. It is so
Yeah.
freaking awesome.
Yeah, and there's a ton of this type of thing on YouTube. That's where we're streaming this from. And there's a ton of different, like, various reproductive structures, or they're called sporocarp. You can even like Google sporocarp spore release video. But yeah, my internet is not at its best. Sometimes when it gets hot, my internet has issues. So yeah, like the box outside expands and contracts, unfortunately.
Dr. Carter, is that sporocarp or sporocarp?
O-carp, like with an O. Like sporocarp. Like spore, O, and then like the fish. Okay, back to our slides. Okay, can you guys see the slides now?
Yes.
Okay, success.
Wait a minute. We're back. Yeah, now we can.
Okay, all right. So
Wait. Sorry. You click something and it goes back to the link to the video, and then you touch something else, the photo comes up.
It's because it's embedded in the presentation. We're good.
Sorry.
Oh, you're fine. You're fine. Okay. All right. So now that we've looked at what happens with mushrooms, let's look at this bottom right photo. So this is actually a reproductive growth. It's actually commonly called a gall, even though it's actually a fungal mass. But it looks kind of like a brown ball of Play-Doh whenever it's developing. And you'll see it kind of wrapped around a cedar tree limb. And, you know, it will literally look like just a brown ball of Play-Doh, someone smushed around it. Now, when it's mature and you have, you know, some nice days of warm, humid conditions, you'll have these orange, sometimes yellowish horns, horn-looking things, they're called horns, emerge out from that ball. And those are called teleal horns, actually. And those are covered in sporulation structures like a mushroom has. So looking at the physiology and where that thing grows, how do you guys think that those spores spread? Water splash or wind?
Wind.
Okay. Excellent. Okay. I was about to say, anyone thinking wind, you are right. So these are really easy examples of how reproductive structure physiology has a role in how those spores are spread. And there's a little note, so y'all have it. Okay, oh, I forgot to mention. And we link that back to diagnosis in terms of, you know, if you have a problem and then you get a diagnosis and you can do some research or have a specialist like myself help you do that research and you better understand how it's spread, then you can better manipulate, say, your environmental conditions to reduce the likelihood of spread. Okay, so some tomato troubles in Texas. I see early blight a lot, like probably one of the most common issues I see. It's caused by a fungus called Alternaria solani, and it favors warm to moderate temps and humidity. So again, that basically fits exactly where you guys are. And this particular disease or pathogen overwinters in crop debris, so old dead leaves laying around, alternate nightshade hosts, so potatoes or weeds, like there are a lot of solanaceous weeds out there. And it can also be seed-borne, so getting clean seed is really important to prevent getting disease problems with this. So again, it also infects potato, and I like to really point that out because in areas that are far south like us and you guys, there are extra risks associated with year-round production because you guys are probably warm enough where you can grow potatoes at least most of the year if it's like in a covered area. So let's say you have tomatoes going in the hot part of the year and you have potatoes growing in like fall, and you have year-round production because you kind of partially protect your crops, you can actually continuously have this problem alternating between those two plants. And the pathogen population will just keep increasing and getting bigger and bigger, and you'll have a bigger and bigger problem. So host range, in this case, is an important thing to consider when you're trying to do things like crop rotations. For prevention and treatment tips, there are some really great resistant varieties like Market Supreme. There's some other ones that have come out recently. Minimize your splashing, optimize air circulation, because again, this loves humidity. And if you can open up that canopy, then it'll help keep things dried out. Remove infected lower leaves to slow the spread. Usually with this, the lower leaves are going to be infected first. Practice really good end of season sanitation. Don't leave plant material laying about. Remove volunteers and those solanaceous alternate hosts. Fungicide applications can be helpful in some cases, but again, especially for homeowners, you have to look at, is it really financially worth it to spend 45 bucks for 8 pounds of tomatoes? And cover your plants with UV absorbing vinyl. And this can actually reduce your disease pressure by around 50%, because this particular fungus actually really needs UV light for it to sporulate. And this is actually linked to the photo on the bottom right here. You can see these kind of advanced looking lesions. And notice you have that alternating light, dark circular pattern. The dark rings are where it's sporulating. And the light part right there is just where the fungus is growing inside the leaf and destroying tissue and consuming it, but it's not actively sporulating. So these are actually diurnal rings. So it's sporulating during the day when it has light and not at night. So if you cover your plants with a UV absorbing vinyl and deny it that UV light, then you can actually reduce your sporulation, and you'll still have some leaf spots that'll look more like on the left, but you won't have extensive sporulation or reproduction and extensive spread. So that's a little fun fact there. And also practice a 2-year rotation just to let any residues, any crop residues left behind fully decompose between crops. Oh, and here again, our spore friends. These are again those classic teardrop shaped spores of Alternaria, and that's how I diagnose it under the microscope. Now, if you have a really, really good hand lens, you might be able to see these, and they'll look like itty bitty little black teardrops in a chain like that stick up off the leaf. All right, powdery mildew is another super common fungal problem that I come across. And it usually comes in as a kind of a pale gray to white kind of cottony substance on the top of leaf. And when you look really closely though, the most common type of spore is what we call conidia. So these are asexually reproduced spores. So they're genetic clones of the parent fungus, and they're clear, even though in mass it looks white. So this is by far the most frequent type of sporulation we have. And these asexual spores are ones that reproduce over and over and over throughout a single season, like we talked about before in the generalized life cycle. Now, this particular pathogen also makes sexually reproduced spores in the form of Cleistothecia. You don't have to worry about remembering that word. Just know they're these little black balls, and they're filled with sexually generated spores. So you have more genetic diversity in these. And these little black balls are filled with spores. And these can be visible with just a so-so hand lens. You don't need a really good high-end one to see them. But these are kind of your hallmark features of powdery mildew. So the thing about powdery mildew is it is usually very species specific. So what infects, or the species that infects like grapes will not infect roses. And the one that infects roses will not infect pumpkins. So generally each species or family of plants will have its own species of powdery mildew. It's very specific. So that's a little different than some of the things we've talked about so far. It overwinters in crop debris and related, so in the same family, plant hosts nearby, often in the same genus. Spores can be blown in from a ways away and also brought or survive on that crop residue. And crop rotation is not very effective due to that, because those spores, like those little clear spores, can be blown in from a ways. Seed treatment is not effective due to the fact that we get spores blown in later in the season. So a lot of times people ask me about that, and it's just not effective. So prevention and management tips. Resistance when available. That's a great way to prevent a problem. This is something I come across a lot in crepe myrtle, like probably the most common problem on crepe myrtle, but there are a lot of really great resistant varieties of crepe myrtle. Sanitation of heavily infected materials, so remove those and dispose of them. You can also try to wait it out if you can. Hot, dry weather inhibits this pathogen. That's part of the disease triangle. The environment becomes unfavorable, so we stop having an epidemic event. Unfortunately, sometimes if it's extremely severe, you can't wait it out before the plant dies. That would happen every year on my zinnias in Arkansas. It was so bad, they would struggle on, but couldn't make it until it got hot enough to resolve the powdery mildew problem. Fungicides are helpful here, preventative and continuous. Like again, on my zinnias, I knew I was going to have a problem every year, so I would just get out ahead of it and do a preventative application. The key here is to stay on schedule. That's very important to avoid the occurrence of fungicide resistance in the fungal population. Some people say potassium bicarb. Again, that's not sodium bicarbonate, which is baking soda. Those are two different things. And that, you know, does seem to help some. Other people have said milk 1 to 10 with water. No, I've not seen great data about that. Feel free to try it and report back. Well, I'm sure we'd all be interested. All right, so on to fungal-like organisms. We're running close on time, but we should be okay if we hustle. All right, so there's a group called oomycetes, and it's a group of organisms that when I was in high school was it was grouped together with fungus. Now it's its own separate group, formally speaking. And that is because even though it looks a lot like a fungus, it does behave differently than a true fungus, like what we just talked about. So these include your downy mildews, like up here on the top right on spinach. That's super common. Pythium, Phytophthora, both these are really common root rots. Earlier we looked at the Pythium blight of pepper. And here on the bottom right, you can see a seedling that has been afflicted with some Pythium, and it's completely killed the seedling. And here on the bottom left, you have Phytophthora root rot. That's these black roots right here on strawberry. And that's a very common issue in Texas strawberry production. And if you were to try to touch that root and grab a hold of it and pull on it just gently, it would completely slide off and just leave the steel or the cortex inside. So that root would be completely dead through and through. Now, oomycetes typically have a very unique means of spore dispersal. They're a little different than fungus. You know, the things we've looked at before have had passive spore dispersal, so like wind and rain, but oomycetes actually have both passive and active spore dispersal. They do make multiple types of spores. Some dispersal is passive, but the ones we're going to really focus on today are active dispersal zoospores. Okay, so again, Phytophthora blight on peppers, super, super common in your area, down in the valley as well. And this pathogen survives in soil, on crop debris, and it may be seedborne. The hallmark symptoms to look for are these chocolatey brown lesions. It often starts from the bottom and works its way up. And on the roots, or I'm sorry, on the fruit, the infection will often have a greasy appearance, especially at the lesion line right there. You can see it looks really water soaked, and you'll often see a lot of fungal, what we would call fungal, even though it's not true fungus, but you'd see a lot of sign, which is actual pathogen tissue right there on the fruit. So we would focus, like if this were my garden, I would focus on future replants with resistant varieties, the use of fungicides. Once I noticed I had a problem in one plant, I would try to use some protectant fungicides on my other plants. Sanitation of infested material and clean seed. So I wouldn't leave old infested peppers like this laying around on the ground, because it will just serve to carry over inoculum to next year. All right, so another common issue that I see is Pythium. This was actually unfortunately a blurry photo. It was one of the first photos I took when I started my job, and I didn't have a great camera yet. But this is actually hydroponically grown wheatgrass, and these are wheatgrass roots. And on the bottom of that wheatgrass mat, you can see that the roots were discolored as this kind of tan color, also kind of yellowish, and there was a lot of fungal sign or pathogen sign. So I picked one of those little roots and I looked at it under the microscope, and here that is. You can see the root with the cortex in the middle, that kind of dark section. And if you look at the red arrows, you can see what look like almost kind of like little Christmas tree ornaments, like little balls embedded in the root plant cells. So zooming in on that, these rectangular shapes are the plant cells of the root, and then you can see these little, what look like balls or Christmas ornaments filled with little globby looking items. So these are actually called zoospores that are inside the oogonia of the reproductive structure. So this is the reproductive structure of Pythium, this little ball right here, and it's filled with zoospores. Okay, so this will play, fingers crossed. Yes, okay. All right, so here is Pythium, or well, this is Phytophthora, but they're similar. This is an oomycete and culture, and here we have zoospores emerging from that reproductive structure. And this is in a thin film of water under a microscope. So these little guys can swim around very much like little tadpoles in a film of water on a microscope slide, or in the soil, for example. And this is real time. They are pretty quick. And this is, again, just off YouTube. You can go watch tons of these videos if you're interested. So these, again, when they're mature, erupt and swim off to go find a new plant host to infect. Okay, so once these do emerge, they don't just, swim about aimlessly. They actually can sense where there are new roots to infect. So they do that through a process called chemotaxis, and they sense chemicals that are coming from roots, especially young root tips, things like hormones and sugars that are released in abundance by root tips. And so just like you can follow your nose to some cookies baking or whatever your dessert of choice is, or brisket, here in Texas, we can all follow our nose to brisket, or what other vegan option you prefer, if that's your cup of tea. But zoospores do a similar activity. So here they're swimming in water. Free water just means enough water to have a thin film. And so here we have a root tip, and these little guys can actually sense nutrients and hormones that leak from this area and follow the increasing strength of that signal until they find the root tip. And once they find the root tip, they go through a process called encysting, and that's ENCYST, not like insist, like put your foot down, but insist, like set up shop. And then they start behaving more like a fungus again. So they'll act like a spore and start germinating a hyphae and penetrate down into the root and start colonizing it. So they're pretty unique in that they kind of act like a or like an animal, hence the term zoospore, and then they revert back to acting more like a true fungus. So they're a little different. So I always like to touch on this for you guys because in terms of okay, I covered that. All right. So in terms of management, though, no. Hold on. 1 second. I'm on the wrong screen. Okay. So in terms of management, though, oomycetes like this have different management needs than a true fungus like powdery mildew, and they also have different fungicides in many cases. So always pay attention to what your diagnosis is, and pay attention to what the fungicide says it's labeled for. So if it says it's labeled for oomycetes or water molds, then it's appropriate to use on something like Pythium or Phytophthora. But if it doesn't include that, then you should not use it on oomycetes because it's probably not effective, and vice versa. Okay, so this is the last section that we have to get through today, diagnostics and exercises. So I'm just going to run through this really quick. You guys are not expected to be diagnosticians at the end of this class. It's just to show you the process so that you can better understand information that I need to do my job. So when you send me stuff, you know what to include. So diagnosis is super important because incorrect management attempts can make problems worse, waste your time and your money, and lead to a lot of frustration. Most fungicides only work on fungi or oomycetes, not bacterial, viral, or abiotic problems. The one exception is copper. It is also labeled for bacterial issues. And also you need to know if you have a diagnosis that involves an insect vector that you need to focus on control, particularly for some viruses and bacteria. And for things like citrus greening, you need to know if a quarantine is going to be important for you. All right, so what are the steps I take to diagnose a problem? First, I ID the host plant. I get some weird stuff sometimes, so I always make sure I know what I'm dealing with. And then I assess the problem. I do research and develop a suspect list, and then I narrow that list of suspects. And again, remember, this is just to give you an idea, so you have insight on the type of info and samples to send me. You don't have to perform under pressure and give a diagnosis for your tests. All right, so ID the host plant. This is an important process of elimination step in that you can automatically rule out a ton of problems that are not an issue on that particular plant host, because not all pathogens infect all plants. You can consult the plant owner, your county agent's great, online resources, botanical keys and field guides are all wonderful sources. The one thing is beware of common names. For example, what we call pigweed here and what we call pigweed in Arkansas are not the same thing. They're in the same family, but not the same species. So just be aware of that. Assess the problem, examine the whole plant top to bottom and the surrounding area, and take a good site history and decide if it's even a disease or is it an injury or disorder. So consider recent weather, soil testing history in terms of nutritional problems, sprays, lightning, deer, and also how long has this problem been going on. Usually diseases are not going to pop up overnight. They're going to be a work in progress type of thing that takes time to develop versus say like a chemical injury will seemingly pop up overnight. Also look at how many plants are affected, a single species or more. How is that area distributed? Looking for patterns can be helpful. And, you know, if you have a problem that is bridging like multiple like groups of plants like monocots and dicots, then it's unlikely that it's a disease. It's probably going to be an abiotic problem. All right, do the research and develop a suspect list. So evaluate those symptoms, look for signs like fungal tissue. That's always a great clue. Reference books and online can be good resources. And I also really consider what's common on the plant host, because usually if it looks like a duck and quacks like a duck, it'll be a duck. Focus on reputable resources. There's some really squirrely things out on the internet, so just make sure you find some good quality material that's reliable. All right, then narrowing that list of suspects, there's a series of books called compendium books that are put out by APS Press. That's like the main professional organization for my field. And these are unfortunately a little pricey, but you can go together with a group of friends and buy a favorite crop like roses if you want. And it has an extensive listing of all or most of the known problems on that particular plant or group of plants. So those can be a really great tool, though, again, they're a little pricey. But these are kind of the gold standard for my field and one of my first go-to things that I use for investigating problems. All right, so let me see. Okay, so how are we on time? Let me see here. Okay, all right, so we're close on time, but I think we can squeeze this in. This is literally like the last thing. All right, so case study 1. Someone brings you leaves, you know, once you're a master gardener, someone brings you this plant and there's a couple different species. They don't tell you what it is, but there's a rough leathery texture on the leaves, and it's widespread on all these different species, and there's a lot of yellowing. You can see that in the picture here. So what's the first thing you're going to do? And the second. So normally in person-to-person class, I put someone on the spot and make them answer, but because we're close on time, I'm just going to give you the answer. It's on your handout anyway. You ID the host. So in this case, it was citrus. Someone brought me some grapefruit and some orange and some Mexican lime. Then I assess the problem, and I take a site history and get a distribution of, or an understanding of the distribution of the problem. So in this case, it was actually severe scale and mite damage. Well, mostly mites, a little bit of scale. So this is one of those kind of trick questions that I include just to be a little mean, to also illustrate that it's really important to ask, and just find out as much of a background as you can. So in this case, the homeowner had never sprayed anything for insect problems, and they had very densely planted citrus, and they just had a really high buildup of mites. So, okay, so case study 2, someone brings you a dying plant, like the whole thing, they pulled it up from the roots, and it has defoliation and severe wilting. What would you ask the person? What would you first do to the plant? And this is what it looked like. So this is what the people brought me. Okay, so the first thing I would ask the person is, what is it? So when I first started my job, I'd never seen sesame before. And this is actually sesame. This is like my second sample ever. So host ID, and then I inspected it top to bottom, and you can see from the outside that we have severe stunting. It doesn't have any branching. It has poor yield set, you know, really poor, unthrifty looking roots. So I also looked at the inside, because that can be a big clue. Remember, we looked at like bacterial wilt earlier, where you split the stem and you have that discoloration. Well, on some other diseases, we also have a similar situation, though a different pattern and different color. But here I had the inside of a healthy stem, and then you can see the lesion on the outside of the stem, and then I split some more stems. And you can see on the inside, we have this kind of grayish, blackish cast of discoloration on the inside in the pith. So this actually had what's called charcoal root rot. And here on the Petri plate picture in the middle, you can see that black fungus growing out on half. That's charcoal rot. And it's very common on a lot of veggies, as well as obviously sesame. And it causes that kind of grayish, blackish discoloration on the inside. Now, we also have this creamy colored fungus growing out on the bottom half of that Petri plate. And this is actually Rhizoctonia root rot. And this is what's causing that deep lesion on the outside of the stem. So this is a case of there's 2 infections going on at the same time. And I think it's forming a disease complex. I need to do some more research to map that out better. But I do see this in tandem a lot. And unfortunately, they have counterintuitive management efforts. And charcoal rot, it loves hot, dry conditions. So we usually recommend people irrigate, and that helps alleviate the problem because it makes the fungus unhappy, if you will. Versus Rhizoctonia really likes it wet. So with this particular producer, if he irrigated to alleviate the charcoal rot, he was exacerbating the Rhizoctonia and vice versa. So that was a little tricky. All right, so that's the end of the formal presentation. I have a home reading section with some bonus slides that include some slides of things that are in my normal longer lectures. I usually do a 4-hour lecture, 3.5 to 4 hours, and some other useful info. So here's the burlwood and things like that. Also at the very end, there's a slide with my digital business card. It has all my contact info. So if you guys have a problem, you can get a hold of me that way.
Oh, what you call burlwood, we call crown gall up north.
Crown gall is going to occur on the crown area of the tree. I mean,
at the flare, yeah.
Yeah, usually, unless it's deliberately inoculated elsewhere, typically.
But that looks the same almost.
Right, like to the naked eye or to the untrained eye, if you will, they do look very similar.
Yes, naked
like
eye,
this
yes.
Yeah, like this one on the top left, I suspect that's a fungal burl.
Okay.
And that, to me, it looks different than crown gall. Like the texture of it looks a little different. It looks a little corkier maybe, versus crown gall looks more like bulbous galls to me.
All right.
But I've been I know that it sounds like very nitpicky and they probably look the same
No,
to everybody, but
I've only had intro level training multiple times, so you get, you know, the kindergarten level again and again and again. It's cool.
well, it's a practice thing, and that's what a lot of this is just when you see it over and over and over, and you see the pictures over and over or samples in person, then you notice those really subtle differences, and it's just practice. That's
And the
all
only
it is.
advanced training I've had are 2 days with Femi and Dr. Ong, and then I got very, very sick. I've had surgery to correct part of it, but I'm still recovering.
Oh, that's rough. Yeah.
Yeah.
Well, I'm trying to put together online advanced training. So it won't be the same as a lab course, but it'll be very photo heavy.
I wasn't able to complete all the field work, and I wasn't expecting field work. So
Yeah. Yeah, it's a struggle.
yeah. Hey y'all, any questions for Dr. Cochran? I had a question earlier. Oh, you were talking about other types of nematodes. Are some of the other parasitic nematodes, do they not form cysts in the roots then?
They do form those kind of the feeding sites, but some of them are migratory, so they form a feeding site and then move on and form another feeding site. That stated, though, most of them will not form those really visible galls. That's why I include root knot nematode in today's lecture, because it forms like visible, like obvious galls, and which is good for class. But
So
like cyst and sting nematode, you pretty much will just need a like nematologist to do a soil count for you or an egg count on your soil and plant sample.
So if I have a bed where there are plants like randomly dying and the roots are extremely depleted, I can't presume that's a root rot?
I would not.
Interesting.
Yeah, I would just send it in to the clinic and see. Terry Wheeler's our nematode specialist. She's great. But the clinic in College Station, they can do a like full analysis and tell you what's what.
Well, this is mostly contributing to the next presentation, but
Okay.
but good to know. I will not propagate misinformation.
Yeah, that's why diagnosis is so important.
I was misinformed about viral variegation, and I've been spreading misinformation for 10 years, and I was corrected last year. Now I'm not propagating misinformation anymore.
There you go.
But you know, we learn as we go. That's